Hello, this is Harsha M, roll number 20E2509 from BSc Biotechnology, first year, and I have come up with a presentation on genetic code. So, genetic code is similar to a secret message we use with some kind of a code to keep the message hidden. As it turns out, decoding messages is also a key step in gene expression, the process in which information from a gene is used to construct a protein. How are the instructions for building a protein encoded in a DNA, and how are they deciphered by the cell? Through this presentation, we'll take a close look at the genetic code, which allows DNA and RNA nucleotide sequences to be translated into the amino acids. This is the contents. First, we'll be looking into genetic code, its definition, discovery, and types, and then we'll move on to the features of genetic code, and then we'll be seeing some hypotheses like the adapter hypothesis, and we'll see how amino acids are attached to the tRNA and the codon anticodon interaction, which leads to wobble hypothesis and different types of mutations. Now, genetic code is a set of rules by which information encoded in genetic material, DNA or RNA, is translated into proteins by living cells. The complete set of correspondences between codons and amino acids is known as a genetic code. The term genetic code was coined by George Gamow. Now, if we look into this diagram, we see that AUG, AUC, UCG, and UAA are the codons which is present in this mRNA, and these are the amino acids that are being translated, and this forms the polypeptide chain. First, let's see how the genetic code was discovered. So, to crack the genetic code, researchers needed to figure out how sequences of nucleotides in a DNA or RNA molecule could encode the sequence of amino acids in a polypeptide. Now, why was this a tricky problem? In one of the simplest codes, each nucleotide in a DNA or RNA molecule might correspond to one amino acid in a polypeptide. However, this code cannot actually work because there are 20 amino acids commonly found in proteins and just 4 nucleotide bases in DNA or RNA. Thus, researchers knew that the code must involve something more complex than a one-to-one matching of nucleotides and amino acids. Here came the triplet hypothesis. In the mid 1950s, the physicist George Gamow extended this line of thinking to deduce that the genetic code was likely composed of triplets of nucleotides. That is, he proposed that a group of 3 successive nucleotides in a gene might code for one amino acid in a polypeptide. Gamow's reasoning was that even a doublet code, that is, 2 nucleotides per amino acid, would not work, as it would allow for only 16 ordered groups of nucleotides. That is, we knew that there are 4 types of bases, and if they were present as doublet code, that would give us 16 amino acids, which is too few to account for the 20 standard amino acids used to build the proteins. A code based on nucleotide triplets, however, seemed promising. It would provide 64 unique sequences of nucleotides. That is, instead of being present as doublet code, if they were present as triplets, then it would give us 64 amino acids, which is actually more than enough to cover the 20 amino acids. Gamma's triplet hypothesis seemed logical and was widely accepted. However, it had not been experimentally proven, and researchers still did not know which triplets of nucleotides corresponded to which amino acids. Here came Marshall Nirenberg. The cracking of the genetic code began in 1961 with work from the American biochemist Marshall Nirenberg. For the first time, Nirenberg and his colleagues were able to identify specific nucleotide triplets that corresponded to particular amino acids. Their success relied on 2 experimental innovations. First, a way to make artificial mRNA molecules with specific known sequences. Second, a system to translate mRNAs into polypeptides outside of a cell, that is, a cell-free system. Nirenberg's system consists of cytoplasm from burst E. coli cells, which contains all of the materials needed for translation. First, Nirenberg synthesized an mRNA molecule consisting of the nucleotide uracil, which was known as the poly-mRNA. When he added these poly-mRNAs to the cell-free system, he found that the polypeptides made consisted exclusively of the amino acid phenylalanine. And because the only triplet in poly-mRNA is UUU, he concluded that UUU might code for phenylalanine. Using this same approach, he was able to show that poly-C mRNA, that is CCC, was translated into polypeptides made exclusively of the amino acid proline, suggesting that the triplet CCC might code for proline. Other researchers, such as the biochemist Hargobind Khurana at University of Wisconsin, extended Nirenberg's experiment by synthesizing artificial mRNAs with more complex sequences. For instance, in one of the experiments, he generated a poly-UC mRNA, that is UCUC, and added it to a cell-free system similar to the Nirenberg's. It was found that the poly-UC mRNA was translated into polypeptides with an alternating pattern of serine and leucine amino acids. These results thus confirm that the genetic code was based on triplets, and today we know that the serine is encoded by UCA and leucine is encoded by CUC. By 1965, Nirenberg, Khurana and their other colleagues had deciphered the entire genetic code through this technique, and they also went on to receive the Nobel Prize in 1968 for their contributions. Now let us see different types of codons that are start, stop and normal codons. Translation always begins at a start codon, which has the sequence AUG and encodes the amino acid methionine in most organisms. Thus, every polypeptide typically starts with methionine, although the initial methionine may be snipped off in later processing steps. A start codon is required to begin translation, but the codon AUG can also appear later in the coding sequence of an mRNA, where it simply specifies the amino acid methionine. Once translation has begun at the start codon, the other codons of the mRNA will be read one by one in the 5' to 3' direction. As each codon is read, the matching amino acid is added to the C terminus of the polypeptide. Translation continues until a stop codon is reached. There are 3 stop codons in the genetic code: UAA, UAG and UGA. Unlike start codons, stop codons don't correspond to an amino acid. Instead, they act as stop signals indicating that the polypeptide is complete and causing it to be released from the ribosome. More nucleotides may appear after the stop codon in the mRNA, but those will not be translated as part of the polypeptide. And here UAA is called as ochre, UAG codon is called as amber, and UGA is called as opal. And the rest of the codons other than the start and the stop codons are called as the normal codons. Now this is the codon table, and here we see that for example, let us consider this box. So we see that in this the first codon in this is C completely, and the second letter here is the U. And in this box we see that the first letter is again C and the second letter here is C completely. So that is how the entire pattern of the table is, and the stop and start codons are also mentioned along with different specific amino acids. We see how an mRNA is getting translated. So the translation begins at the start codon AUG, and amino acids are being added It begins from the N terminus, and the last terminus known as the C terminus. And once the stop codon here UAA is reached, it gives a stop signal, and the mRNA stops being translated, and the polypeptide is formed, and later it gets snipped off to form a protein. Now, the start codon is very critical because it determines where translation will begin on the mRNA. Most importantly, the position of the start codon determines the reading frame, or how the mRNA sequence is divided up into groups of 3 nucleotides inside the ribosome. As shown in this diagram, same sequence of nucleotides can encode completely different polypeptides depending on the frame that is being read. The start codon determines which frame is chosen and thus ensures that the correct polypeptide is produced. Now, for example, let us first consider the frame 1. We see that the translation begins at U and we get the first amino acid serine. Now we see that no proper protein is formed here because the second set of the codon is UGA, which is a stop codon, and thus we don't get any proper protein because the mRNA gets terminated, or the translation stops very soon. So no proper protein is formed in the first case. Let's consider the frame 2. Here we see that the translation begins at C, and we see that different amino acids are getting translated. But the problem here is that there is no stop signal here to stop this translation process, and this continues to form an improper protein. So even this is not a desirable case. Now let us consider the third frame. Here we see that the translation begins at A, and the first amino acid form is methionine. So the codon used is AUG, which is our start codon, and we see that amino acids are getting translated, and it reaches a codon UAA, which is a stop codon, and when it reaches there, the stop signal terminates the process and we get a proper protein. So in this frame 3 we get a proper protein. So through this diagram they indicate that the start codon's position ensures that this proper frame is chosen. That is, a start codon is very important for us to get a proper protein. Now, there are a lot of properties associated with the genetic code, such as degenerate but unambiguous. We know that each amino acid is represented by a triplet sequence. Excluding all the start and stop codon, there are 61 codons for 20 amino acids. So, therefore, one amino acid has more than one codon, and the code is referred to as degenerate. Now, for example, let us consider arginine. This can be coded by CGU, CGC, CGA, and CGG. Although an amino acid may have more than one codon, each codon specifies only one amino acid. Thus, the genetic code is unambiguous. Codons that designate the same amino acid are called synonyms. Methionine and tryptophan each have only one codon. The second property is that the genetic code is nearly universal. With some minor exceptions, all living organisms on Earth use the same genetic code. This means that the codons specifying the 20 amino acids in our cells are the same as those used by the bacteria living in places like the hydrothermal vents at the bottom of the Pacific Ocean. Even in organisms that don't use the standard code, the differences are relatively small, such as a change in the amino acid encoded by a particular codon. A genetic code shared by diverse organisms provides important evidence for the common origin of life on Earth. That is, many species on Earth today likely are to be evolved from an ancestral organism in which the genetic code was already present. And because the code is essential to the function of cells, it would tend to remain unchanged in species across generation, as individuals with significant changes might be unable to survive. So, this type of evolutionary process can explain the remarkable similarity of the genetic code across present day organisms. But there are a few exceptions to the universality of the genetic code, which are found in human mitochondria, like UGA codes for tryptophan instead of a stop codon, AUA codes for methionine instead of isoleucine, and CUA codes for threonine instead of leucine. The next property is that the code is non-overlapping. So during translation, the code is read sequentially without spacer basis, taken three at a time. Now, for example, let us consider this sequence. In this sequence, this is read as AUG, this is read as CUA, this is read as GAC, and this is read as UUU. We see that there are no commas or no spaces between them. That is, the code is non-overlapping and without punctuation. Finally, the code has polarity. The code has a definite direction for reading of message, which is referred to as polarity. Reading of message from left to right and right to left will specify for different amino acids. For example, UUG, which codes for leucine, where it is read from right to left, it gives amino acid, it forms a codon GUU, which actually forms, codes for valine. So we see that in these, when the codon is read from right to left and left to right, we actually get two different amino acids. So it is really important to read the genetic code in one particular direction, that is 5' to 3'. Next is a hypothesis called the adapter hypothesis. In 1950, Francis Crick proposed that the small molecules made out of RNA could help codons of mRNA interact with specific amino acids in order to create polypeptides called proteins. That is, codons of mRNA use certain small molecules made out of RNA to interact with the specific amino acids to form the proteins. It was an explanation to how genetic information stored in RNA could be converted into proteins. That is, it was an explanation to our central dogma. To put it simply, it was hypothesized that there was an intermediary called an adapter molecule that could help adapt the mRNA into amino acids to form proteins. This adapter molecule was later discovered to be tRNA. Now, in this diagram, we see that the tRNA brings amino acid to the mRNA and brings it to the ribosome at the A site. It links its amino acid at the P site and exits at the E site. So the definition of an adapter is a device for connecting pieces of equipments that cannot be connected directly. The tRNA in this case acts as an adapter between the mRNA and the amino acids. The anticodons on the tRNA determines the appropriate amino acid. This anticodon base pairs with the mRNA sequence and then deposits its amino acid into the growing polypeptide chain. Next is the codon-anticodon interaction. The codon of the mRNA is recognized by the anticodon of tRNA. They pair with each other in antiparallel direction, that is, 5' to 3' of the mRNA interacts with 3' to 5' of the tRNA. The usual conventional complementarity base pairing of AUCG occurs between the first two bases of anticodon. The third base of the codon is rather lenient or flexible with regard to the complementary base. The anticodon region of tRNA consists of 7 nucleotides and it recognizes 3-letter codon in mRNA. Now, these 2 points bring in a lot of questions like how could this be possible considering the Watson-Crick complementary base pairing principle? This could be explained well through the wobble hypothesis. Now, what is this wobble hypothesis about? We know that in translation, one codon interacts with one anticodon to give one amino acid. And then we know that there are 20 amino acids, hence will surely require 20 tRNAs. But there are 61 codons for 20 amino acids, and the number of tRNA is far less, which is due to wobbling. Crick postulated the wobble hypothesis to account for the degeneracy of genetic code. According to this hypothesis, the first two bases of the codon pair, according to the normal base pairing rules, pairs with the last two bases of the anticodon. Base pairing at the third position of a codon is wobble. Okay. If we look into the codon table, we'll find that in every section of the codon table, the different types of codons for one particular amino acid. In that, the first two bases remains the same and the third base always differs. For example, UCA, UCU and UCG. All of these codes for serine. So here we see that in all these three, the first two bases are conserved and only the third base slightly varies. As said earlier, there are many amino acids in our body, and to have a separate tRNA molecule for all the amino acid is kind of very much complicated. So sometimes in our body, one or two different tRNA will have different varieties of amino acids coming due to the position of a particular codon, because each codon represents a particular amino acid, but slight variation of the codon in the third position doesn't change the amino acid, and that particular codon also codes for the same amino acid as seen in this case of serine. It explains the existence of multiple codons for a single amino acid. It is a phenomenon in which a single tRNA can recognize more than one codon. Okay, so to put it simply, wobble base pairing means the base pairing between RNA molecule which does not follow the general rule of Watson-Crick base pairing. We know that in Watson-Crick base pairing, A forms double bond with T or U and G forms triple bond with C. But in wobble base pairing, we observe these are the anticodons in the tRNA, and these are the codons in the mRNA. So in case of adenine in the anticodon, the usual way of pairing is with uracil as the codon, and here C pairs with guanosine. These two types of pairing, these are strong pairing, and that follows the Watson-Crick pairing rules. But here we have G guanosine, and that pairs with C based on the common base pairing rule, but this G can also pair with uracil U, though not as good as it pairs with C, but it definitely can still pair with uracil. Now here again, uracil can pair with adenine as well as guanosine. The last one is I. It is hypoxanthine, a derivative of inosine bases, hence the letter I. This inosine can pair with adenine, cytosine, and uracil. This hypoxanthine is often found in RNA than it is found in DNA. That's why wobble pairing is mostly found in RNA, because most of the wobble pairing occurs between inosine and either A, C, or U. So these are the wobble base pairs. Now this is an example of wobble hypothesis. This is the case of normal pairing for leucine, and this is the wobble pairing. Here we see that G forms bond with U, uracil, and in normal it forms with C, cytosine. But both gives us the amino acid, the end product, leucine. Moving on to mutations, there are two types of mutations. The first one being point mutation. Under it, we have another three types: silent, nonsense, and missense. Let's look at the mRNA level. First, the silent. In this, there is a single nucleotide change, that is G to A. But same amino acid is incorporated, that is, in both the cases lysine is formed, thus the mutation goes unnoticed. Next is nonsense. In this, there is a single nucleotide change from A to U. Stop codon is generated, so we see that UAG is a stop codon, so this mutation leads to form a stop codon, and thus the translation gets terminated. So premature termination of chain may at times be incompatible for life. So next is the missense. In both the cases, there is a single nucleotide change from A to G or A to C. Different amino acid gets incorporated, and this mutation leads to loss of functional capacity of the protein. Now the second type of the mutation is frame shift mutation. In this, the insertion or deletion, that is removal of a base, can alter the reading frame with the resultant incorporation of different amino acids, as we can see it in this case. In deletion, we see a different type of amino acid chain being formed, and in this, in insertion, we see a different type of amino acid chain being formed. So we have discussed about genetic code, its features, hypothesis like adapter hypothesis, wobble hypothesis, and finally we have also covered about different types of mutations. So now that marks the end of the presentation. Thank you.